Hello, good evening, everyone. Hello, good evening, everyone. Welcome to the week 5 of NPTEL live session. So, as you know, this problem solving session is held every Monday from 6 p.m. to 8 p.m. With the same shared link. Um, so, I'll forego all these formalities because there are not much people joining here. So, Harshita, if there is any case where there is network disruption from my side, please stay online. I'll try to join as soon as possible. And if you have any question, you can unmute yourself and ask it directly. If not, if you don't want to stop the flow of the class, you can probably just write the question on chat box as the class progress. This is a a live recorded session, and the link will be shared to you later. And yeah, so this week in class, these following topics were discussed. Um, Went more in details with nucleic acid as drug targets and some miscellaneous drug targets. He also discussed some of the targets and some related topics. He discussed some mechanisms in biological chemistry, which was divided into part one, part two, and part three. You might have seen those in, your video lessons. So, without any further ado, I'll start with the questions. These questions again, these questions are framed in a way that this gives you an idea of how to solve the questions in your assignment. You know, let's see the first question. Which of the following terms best describes the programmed cell death mechanism that eliminates damaged or unwanted cells? The first option is necrosis, second autophagy, C apoptosis, and D pyroptosis. The correct answer is apoptosis. This is the definition of apoptosis. So let's see what apoptosis is. It's actually a tightly regulated process that removes damaged or unneeded cells without causing inflammation. It involves capsase activation and DNA fragmentation. Now, apoptosis is, you know, so our body has this regulated function of how cell divides, cell grows, and how cells are like programmed to die. Apoptosis is that function which you know is very tightly regulated by this enzyme, as you can see, capsase. And whenever some cells are like not working in their optimal way, the way they should, they are like, you know tuned to the effect that they are removed, and they are basically crushed and removed. What is necrosis? Let's see why the other options are not correct. The first one was necrosis. Necrosis is uncontrolled cell death. So the keyword being uncontrolled. here. So, necrosis is uncontrolled cell death that results from injury or infection and it causes inflammation. So, the keyword here should be uncontrolled cell death and causing inflammation. In case of autophagy, it's a survival mechanism where cells degrade and recycle their own components and not a programmed form of cell death, unlike apoptosis. Apoptosis is controlled programmed cell death that removes damaged cells without causing inflammation. So you can see the difference between necrosis as uncontrolled, and also it causes inflammation, whereas in case of apoptosis, it is controlled, programmed, and it does not cause any inflammation. In pyroptosis, you see it's a programmed cell death, but is often triggered by infections. Now, let's go to the next question. Which of the following structure belongs to which of the oh which of the following structure belongs to quinoline antibiotic? What is the primary mode of action? A. It inhibits bacterial ribosomes. B. Blocks DNA guidase and topoisomerase 4. C. Inhibits folic acid synthesis. And D. Disrupts bacterial cell wall synthesis. So, can anyone tell me what the structure is? Like, obviously, it belongs to quinoline family, but what is the structure? You can see there's F here. Yes, you're right. Yeah, yeah, it is. But do you know what the structure is? Yeah, perfect. The sixth position of F, like it indicates it's fluoroquine. Perfect. And the correct answer is B. It blocks DNA guidase and topoisomerase 4. So, quinolones and fluoroquinolones inhibit DNA guidase and topoisomerase 4, preventing DNA replication and transcription, often leading to cell bacterial death. Let's discuss why the other options are not correct. The first option was inhibits bacterial ribosomes. This is incorrect because the target of quinolines are not ribosomes. There are certain class of antibiotics like tetracyclines and aminoglycosidase that target ribosomes. The second option, which was the correct option, was it blocks DNA guidase and topoisomerase 4, you know, and that's how it prevents DNA replication in bacteria. The third option was inhibits folic acid synthesis. Again, quinoline does not do that. There's a class of antibiotics called sulfonamides. They disrupt folic acid synthesis. The D option was disrupts bacterial cell wall synthesis. Again, beta lactam is a class of antibiotics that target the cell wall in that disrupts basically the bacterial cell wall synthesis. And yeah, so DNA, just to recapitulate, DNA guidase and topoisomerase 4 are the enzymes that help in DNA supercoiling. If you remember from last to last session where we discussed the replication of DNA, that's where we discussed supercoiling and replication. So the targets of quinolines are like DNA guidase and topoisomerase 4, like ciprofloxacin. Other The third question is the primary target of taxane, a class of anti-cancer drug, is A. DNA polymerase, B. Microtubules, C. Ribosomes, D. Mitochondria. I think there's some background voice coming. Yeah, perfect. You can unmute yourself while answering, but it would be easier just for the sake of recording, nothing else. Yes. So, what should be the answer? The primary target of Texanes. Yeah, it's actually microtubules. So, Texanes, these are a class of so sir actually discussed um, this class of antibiotics. So not antibiotics, anti-cancer drugs. So they basically stabilize microtubules and prevent their depolymerization. So there's a stage in mitosis where uh, there's polymerization and depolymerization of tubulin, and basically this class of drugs, it stabilizes microtubules, and it prevents their depolymerization, thereby inhibiting mitotic spindle formation and arresting cell division in cancer cells. The DNA polymerase obviously is inhibited by drugs like, you know, gentamicin. No, no, no. Yeah, forgive me, that's not the correct drug. I was confused while looking at the example. But yeah, the target of texanes are microtubules, not DNA polymerase or ribosome or mitochondria. Let's go to the next question. Which of the following enzyme uses pyroxido- py- pyro- pyridoxal phosphate (PLP) as coenzyme? A. DNA polymerase. B. Transaminases. C. ATP synthetase. And D. Helicase. Yeah, so just a minute. I would like to stop the recording and like probably. Yes, that's the correct answer. These coenzymes are involved in amino acid metabolism, particularly in transamination reactions catalyzed by aminotransferases, DNA polymerase, ATP synthase, and helicase. These are the three other enzymes involved in replication and ATP, you know, synthesis. They are not its target. Let's go to the fifth question. What is the primary mechanism by which aminoglycosidase antibiotics exert their antibacterial effects? A. Inhibit DNA synthesis. B. Inhibit cell wall synthesis. C. Disrupt bacterial protein synthesis. And D. Interfere with folate metabolism. The correct answer is C. It basically disrupts bacterial bacterial protein synthesis. Aminoglycosidase bind to the 30S ribosomal subunit, causing misreading of mRNA and leading to defective protein synthesis, ultimately killing bacteria. The option A and B are incorrect because obviously cell wall synthesis is targeted by beta lactams, and DNA synthesis is inhibited by quinoline, class of antibiotics called quinolines. The third, folate metabolism is targeted by sulfonamides and aminoglycosides. They aminoglycosides they disrupt bacterial protein synthesis. Let's see the next question. What is the key role of beta lactam ring in penicillin antibiotics? A. It binds to bacterial DNA, preventing replication. B. It inhibits transpeptidase enzymes involved in peptidoglycan cross linking. C. It interferes with ribosomal function. D. It enhances antibiotic solubility in water. Yes, that's the correct answer. So penicillin, they inhibit transpeptidase enzymes involved in peptidoglycan cross-linking. The first option is incorrect because penicillins do not interact with DNA, and they don't interact. Their target is not ribosomes, and it does not enhance antibiotic solubility. It just you know targets the enzyme that is responsible for cross-linking the peptidoglycan for cell wall synthesis, and ultimately that leads to cell lysis and death. The mechanism by which fluoroquinoline achieve bacterial cell death involves: A. Disrupting ribosomal function. B. Inhibiting bacterial RNA polymerase. C. Blocking topoisomerase and DNA gyrase activity. And D. Inhibiting folate synthesis. We just discussed about this class of antibiotics. Uh, they, their primary. Is there anyone willing to answer? Yeah, correct, sir. That's right. Yes, the correct answer is C, blocking topoisomerase and DNA gyrase activity. So, fluoroquinolines such as ciprofloxacin prevent bacterial DNA replication by inhibiting DNA gyrase and topoisomerase 4, leading to bacterial cell death. So, again, because its target is not ribosome or any of the ribosomal function, that's that's a class of antibiotics called tetracycline. They target ribosomes, and yeah, so DNA gyrase, they target DNA gyrase and topoisomerase 4 are the main targets of fluoroquinolines. Which of the following drug is an intercalating agent used in cancer chemotherapy? Why not cisplatin? Cisplatin is also planar, and it also goes and settles between the basis of DNA. Why not cisplatin and why doxorubicin? Would anyone like to answer? Unmute themselves and like answer to this question. Obviously, the correct answer is doxorubicin. We can see cisplatin is not the correct answer because cisplatin forms DNA crosslinks but does not intercalate despite being a planar molecule. Number 9. Which class of antibiotics act by inhibiting bacterial folic acid synthesis? You have the four options. If you remember, we just discussed this. So the correct answer is C, sulfonamides. They basically inhibit the bacterial folic acid synthesis, whereas beta lactams they target cell wall synthesis, tetracyclines they target ribosomes, microlids they target bacterial ribosomes and not folic acid synthesis. What is the primary function of mRNA in protein synthesis? A. It serves as a catalyst for the reaction. B. It provides the genetic code for protein synthesis. C. It forms the ribosomal subunits. And D. It brings amino acid to the ribosomes. So, if you remember, we discussed translation in the class where we discussed replication, transcription, and different various other processes. So, try to remember that. The correct answer is B. It provides genetic code for protein synthesis. mRNA is a crucial intermediary in the central dogma of molecular biology. It transcribes the genetic information from DNA and carries it to ribosomes where proteins are synthesized. It does not serve as a catalyst for the reaction because it's that's the work of different ribosomal uh, protein. That's not exactly, yeah. Uh, Tanmay, I think you should ask this to NPTEL authorities. I'm not sure how the working. Yeah, yeah, yeah. I don't know why that happens. That it shows for me as well, despite going through the video lectures. I'm not sure how that's happening. You can just mail it to NPTEL people. They'll take care of it probably. I don't have any authority over how the NPTEL system works. I just make slides and take classes. I'm sorry I cannot help you there. Um, the subunits are formed by ribosomal mRNA and not mRNA. So again, the third option is not correct. the D option was it brings amino acid to ribosomes. No, if you remember correctly, it's TR. Uh, I can try. I'll try to mail them, but at this point, anyone's voice is just as good as mine. But sure, I can mail this your concerns to them, and we can see how that works. Hopefully, everything will work out fine from the next week. So yeah, but I've raised this issue already in the forum. Yeah, I think forum things are mainly taken care by TAs and not NPTEL authorities. But yeah, sure, I'll try and I'll mail them and let you know how they respond. I mean, if you'd like to unmute yourself, does it affect your marking system? So if the progress is not recorded properly, would that affect your marks? Hmm. Okay, so you have to like mm, complete everything in order to get good comments. Okay, okay. Yeah, sure. I'll try to do what I can. Uh, in the forum. Okay, okay. Sure, sure. Yeah, I'll try to raise this with ma'am, and hopefully she'll take care of it. I'm sorry. Yeah, I'm not involved in. Yeah. No, no, it's okay. I'll try to do my best here, and I'll keep you guys updated. It's okay. So let's go to the next question. Which of the following is not a function of DNA polymerase? A. Synthesizing a new DNA strand. B. Proofreading and correcting errors during replication. C. Ligating Okazaki fragments together. And D. Replacing RNA primers with DNA. Not a function of DNA polymerase. It's because the main topic today is nucleic acid. There are many questions that might seem like repetitive. It's just trying to be very thorough, and it's kind of revision. So try to answer these questions. We'll get over it. So yeah. So can you mute yourself? I think there's some background noise coming, and thank you. So the correct answer is C. Ligating Okazaki fragments together. DNA polymerase extends DNA strands and ensures replication fidelity, that means with correctness. But ligase is responsible for sealing the backbone between Okazaki fragments. Okazaki fragments are the discontinuous fragments, and basically ligase is the one that makes it continuous, it joins them. So option A was synthesizing a new DNA strand. This is incorrect. Sorry? Sorry, is there anyone trying to talk to me? Uh, Dr. Mahesh? Dr. Mahesh? Would you please mute yourself? Can you please mute yourself? Hello? Sir, can you please mute yourself? Thank you. So, the option A was synthesizing a new DNA strand. This is incorrect because DNA polymerase builds new strands of DNA by adding nucleotides. Mind you, the question is asking is not a function of DNA polymerase. So when I say incorrect, that means it actually, the DNA polymerase actually does this work. So number B, proofreading and correcting errors during replication. Actually, DNA polymerase does that. It DNA polymerase has exonuclease activity for error correction. That's like proofreading, making sure correct base pairs with the other base. Ligating Okazaki fragments, DNA ligase does this work, not DNA polymerase. DNA ligase joins the Okazaki fragments on the lagging strand. D. Replacing RNA primer with DNA. Again, DNA polymerase does this work as well. Let's move on to the next question. Which is the primary function of rRNA? It stores genetic information. It carries amino acid to the ribosome. It catalyzes peptide bond formation during translation. Or D. It serves as template for protein synthesis. Yeah, the correct answer is C. It catalyzes peptide bond formation during translation. So, if you remember the process of translation, you remember even in tRNA it had like specific sites of from where it comes, and there's a bond where there's a site where they form peptide bonds between each other. So that is catalyzed by rRNA. So the first option was it stores genetic information. That's not correct. DNA stores the genetic information. The second option was it carries amino acids to the ribosome. That's done by transfer rRNA. C, it catalyzes peptide bond formation during translation. That's true. rRNA plays a catalytic role in forming peptide bonds, acting as ribozyme. The last option was it serve as a template for protein synthesis. That's not true because mRNA serves as the template for protein synthesis, not rRNA. Which enzyme is responsible for unwinding the DNA helix during replication? A. DNA polymerase. B. Helicase. C. Ligase. D. Topoisomerase. So the correct answer is B. You're right. Helicase enzyme. Helicase is essential for replication as it separates DNA strands, allowing polymerases to access single-stranded template for the synthesis. So the option A was DNA polymerase. It helps in synthesis of the complementary base pair, like complementary strand of DNA, but it is not involved in unwinding helicase. It unwinds double-stranded DNA, basically, brings it when and unwinds the double-stranded DNA. Uh, by breaking hydrogen bonds. Obviously, ligase, again, it joins, as the word suggests, it joins the DNA fragments. And topoisomerase reduces supercoiling but is not involved in unwinding of DNA. The next question is which of the following is not a component of a nucleotide? A nitrogenous base, a phosphate group, a ribose or deoxyribose sugar, or an amino group. Yes, that's the correct answer. So, amino group is obviously not a component of nucleotide. A nucleotide consists of a nitrogenous base, a pentose sugar, and a phosphate group. Amino groups are part of nitrogenous bases, but they are not part of a nucleotide component. Let's go to the next question. What is the role of restriction enzyme in molecular biology? A. They cut DNA at specific sequences. B. They synthesize new DNA strands. C. They join DNA fragments. D. They repair mismatched DNA bases. Yes, that's the correct answer. They are known as molecular scissors as well. They cut DNA at specific sequences, and hence are used in recombinant DNA technology. They do not synthesize DNA strands. That's done by DNA polymerase. DNA fragments are joined by ligases, and uh, you know, obviously they do not repair mismatched DNA bases. Let's move on to the next question. What is the function of tRNA during translation? A. It carries amino acid to the ribosome. It forms peptide bonds between amino acid. C. It transcribes genetic information from DNA. Or D. It unwinds mRNA. Yes, again, both of you are correct. It carries amino acids to the ribosome. tRNA has an anticodon that pairs with mRNA codons and carries specific amino acid to the ribosome, ensuring correct protein assembly. It does not form peptide bonds between amino acid because that's catalyzed by rRNA, and genetic information is transcribed by mRNA, not tRNA, and it's not involved in unwinding of mRNA. Helicase unwinds DNA, but there's because RNA is single stranded, that needs no enzyme to unwind during translation. The next question is which of the following statement about DNA replication is false? A. It occurs in 5 to 3 direction. It's semi-conservative. Both DNA strands are synthesized continuously. And D. Helicase is required to unwind the DNA helix. Yeah, again, both of you are correct. Both strands of DNA are not synthesized continuously. One is discontinuous, the lagging strand is discontinuous, called Okazaki fragments. The replication is semi conservative and directional from 5 to 3 but only leading strand is synthesized continuously, while lagging strands are synthesized in fragments, which is later ligated by ligase enzyme. So when I write incorrect again, the question is which of the statement is false. So incorrect means this is actually correct with regard to DNA replication. So it does occur from 5 to 3 direction, and it is semi conservative again. And obviously one of the lagging strand is not synthesized continuously, and helicase is required to unwind DNA ligase. So that's not correct either. The next question: what is the function of telomerase? A. It repairs damaged DNA. B. It extends the ends of linear chromosomes. C. It synthesizes new DNA strands. Or D. It removes RNA primer. Telomerase. So the interesting fact about telomerase is like when we are born, I think so. So as we grow older, the telomerase starts shrinking, and yeah. So yes, the correct answer is B. Telomerase adds protective sequence to chromosomes, and you can see in the structure of chromosome there is like a bulb-like thing added, drawn. So that is telomerase. So telomerase adds protective sequence to chromosome ends, preventing degradation and maintaining genomic stability in dividing cells. It does not repair damaged DNA. DNA repair enzymes fix DNA damage, and telomerase has nothing to do with it. Uh, the synthesis of new strand of DNA is done by DNA polymerase, and it does not remove RNA primer. DNA polymerase does that. Let's move on to the next question. What type of bond links nucleotide together in a DNA strand? A. Hydrogen bond. B. Peptide bond. C. Phosphodiester bond. Or D. Glycosidic bond. Then the type of bond nucleotide together, like not the bases, but the nucleotides. DNA strand. DNA, yeah, the backbone of DNA. The correct answer is phosphodiester bond. It, this they basically um, provide the back that's how the backbone of the DNA is made. So they provide structural integrity to the structure of DNA and RNA by connecting nucleotides through phosphate linkages. So you see the sugar, the OH bond from the OH from sugar binds with the phosphate group, and it forms the phosphodiester bond. The hydrogen bonds are between the complementary ATGC nitrogenous bases, and peptide bonds are not there in DNA. So the next question: which of the following antibiotics was widely used during World War II for treating bacterial infections? A. Erythromycin. B. Chloramphenicol. C. Proflavin. Or D. Rifampicin. The correct answer is C. Proflavin. It's a synthetic derivative, a synthetic ac- acridine derivative that is used as antiseptic, that was used as an antiseptic for treating wounds during World War II. It integrates into bacterial DNA, disrupting replication. Other antibiotics like erythromycin and chloramphenicol, the option A and B, were discovered after World War II and were not widely used at that time. Erythromycin is a microlid antibiotic, and again, these two were discovered after World War II. Rifampicin was discovered even later and is used to treat tuberculosis. Which type of bonds help stabilize the secondary structure of DNA? Now, mind you, not primary structure, secondary structure of DNA. So the options are ionic bond only, B hydrogen bonds only, C van der Waal interactions only, and the fourth is hydrogen bonds and van der Waal interaction. Yeah, okay. I mean, so you have to be careful when the option states like only. Just try to consider all the options. The one that has like that seems to be most suitable, try to pick that answer. Most of the time in MCQs, not all the options are correct, but we have to select the one that's like more suitable, more correct. Let's say the correct option is D, hydrogen bond as well as van der Waal interactions. The secondary structure of DNA is stabilized by hydrogen bonds between complementary base pairs AT and GC, and van der Waal interactions, also called backstacking, that stabilizes adjacent bases. You know, you see their bicyclic and monocyclic structure. There is like pi-pi interaction and that stacks upon each other. That's a pi-pi stacking that makes the secondary structure even more stable. So ionic bonds, no, DNA is primarily stabilized by hydrogen bonds, not ionic bonds. Hydrogen bond only. So we know there is hydrogen bond between the complementary base pairs. So, but it is not only the only force responsible for stability. We have to consider the pi-pi stacking as well. Van der Waal interaction contributes, but is not only responsible for the stability. Hydrogen bonds as well as van der Waal interactions, they both are the correct answer. Next question is, what type of interaction occurs between aromatic rings in DNA base stacking? A. Covalent bonding, B. Electrostatic attraction, C. Hydrophobic interaction, D. Disulfide bonds. So we just discussed the pi-pi interaction between the uh, rings of the nitrogenous bases, purine and pyrimidine, pyrimidine and purine. So ATGC. So once they have like hydrogen bonds between them, like 3 and 2, they are stacked in such a position that that alpha helix, it it takes its position, and they have this hydrophobic interaction that we call pi-pi interaction and pi stacking. So the correct answer is C, hydrophobic interaction. Base stacking occurs due to the hydrophobic interaction among aromatic nitrogenous bases, which minimize exposure to water. Obviously, it's hydrophobic. Mm. These interactions stabilize the DNA helix by reducing entropy. Covalent bonds hold the nucleotide together but do not contribute to stacking. So that's why option A, covalent bonding, it's incorrect. B, electrostatic attraction. No, actually, the negatively charged phosphate backbone, they basically repel. They do not attract. There is no electrostatic attraction there. The hydrophobic interactions, obviously, the pi stacking between the nitrogenous bond and the third disulfide bonds. It occurs in protein, not in DNA. What is the structural characteristic of acridine dyes like proflavin? A. They contain a peptide backbone. C. They have a polycyclic planar structure. C. They form ionic bonds with DNA bases. And D. They act as competitive enzyme inhibitors. A structural characteristic. They're obviously planar and polycyclic, you know. They have like three rings, and in between there is nitrogen ring, and they have like two NH2 NH2 as well. So the structural uh, characteristic would be planar and polycyclic ring that insert between they basically intercalate, and they do not rely in any hydrogen bonding or enzyme inhibition for activity. They do not contain peptide backbone. Yes, sir. Amide. Yeah, I know. Yeah, but exactly. But the question here is structural characteristic. I'm not talking only about proflavin. Uh, they're basically about the dyes, and in most of the cases, they are not charged. So once they are charged, obviously, like in case of particularly in case of proflavin, it's charged, and once it's charged, it forms bond with bases. Yes, sir. Yes, sir. Yeah, yeah, I know. That's why I try to put all the options and clarify. I can understand this is not a very good question in that sense, but we are talking about the emphasis is mainly on the dies. But yes, I understand your point as well. So, but in general, if we are putting emphasis there and we're not talking about the charged thing, yeah, I'll remove this question when I share the PPT. I think it will just create confusion. So I'll remove this question before sharing the PPT. Yeah, yeah, yeah. I understand, but I don't want to create any pressure. This is not adding anything to it. I understand your point and I'll remove that. Which structural feature of DNA allows it to carry genetic information? Double helix structure, single stranded nature, presence of disulfide bridges, or D, presence of uracil. Yes, that's the correct answer. Double helix structure provides structural stability and allows semi conservative nature of the replication, ensuring the faithful transmission of genetic information. So the correct answer is double helix structure. Single stranded nature, obviously DNA is a double stranded, single stranded is RNA. So that's not the correct answer. And RNA, it's so DNA is responsible for carrying you know, genetic information. And C, presence of disulfide bridges. No, it's found in protein, not in DNA. And presence of uracil. Again, uracil is found in RNA, not DNA. So all the other options are wrong. The next question is, what is the role of DNA intercalating agents? A, they replace nucleotide bases. B, they distort DNA structure and inhibit replication. C, they act as ribozyme. D, they enhance transcription efficiency. DNA intercalating agents. So intercalating agents, they go and, you know, they place themselves between the nitrogenous bases. So the correct answer is B. They distort DNA structure and inhibit replication. Intercalating agents such as acidine dyes insert themselves between the nitrogenous base pairs because generally intercalating um, agents are planar in structure. So they go and they place themselves between the nitrogenous bases, and that causes distortions, and that distortion basically stops replication and hence transcription. They do not Intercalating agents do not replace nucleotide bases, and they do not act as a ribozyme. Ribozyme are RNAs, RNA molecule, rRNA, that have like catalytic property, and they do not enhance transcription efficiencies. They actually inhibit a transcription rather than enhance. Which of the following is a function of nucleotide in biological systems? A, only serves as building blocks for DNA. B, act as energy carriers in cells. C, only participate in RNA synthesis. And D, function as structural components of proteins. You know, function of nucleotide. The correct answer is B, act as energy carriers in the cells. Nucleotides such as ATP and GATP function as energy carriers, and they are also precursor of DNA and RNA synthesis. So they have multiple functions beyond the synthesis of DNA, beyond being the unit for DNA structure. So yeah, we discussed enough now about nucleotides. What is the primary cause of DNA denaturation? Strong acid or base, high salt concentration, presence of RNA polymerase, or the DNA ligase activity? So, DNA does not affect the primary structure. The correct answer is strong acid or base. The denaturation happens when the hydrogen bonds break, separating the strands. So, DNA strand, which was earlier double helix, becomes single helix. That is caused in presence of strong acid. That can be caused in presence of like strong acid and base, and with by other factors like heat, extreme pH, or chemical denaturants. So, yeah, uh, the one thing that can affect among all these things, the one thing that can lead to high heat and extreme pH is strong acid and base. Salts can actually stabilize DNA rather than denature it. And presence of RNA polymerase, RNA polymerase transcribe DNA but does not denature it. And DNA's ligase activity is to join DNA fragments and not denature DNA. So the correct answer is A, strong acid or base. The next question is, what is the structural feature that makes RNA less stable than DNA? A, presence of ribose instead of deoxyribose. B, double stranded nature of RNA. C, absence of phosphate backbone. Or D, presence of thymine instead of uracil. So mind you, it's talking about how RNA is less stable than DNA. So, double-stranded nature of RNA. RNA is, hmm, sorry. So the correct answer is A, presence of ribose instead of deoxyribose. RNA has ribose sugar which contains additional OH at 2 dash position, making it more susceptible to hydrolysis compared to DNA. Double-stranded nature is of DNA, obviously. That's that's a very tricky option there. So because we are so used, even while reading the options, my first instinct was to say double-stranded nature of DNA, but the option is double-stranded nature of RNA. That's why it's wrong. Absence of phosphate backbone. No, RNA, like DNA has phosphate bond. They both have the same backbone. And presence of thymine instead of uracil. Uracil is present in RNA, not in DNA. The next question is, which of the following is function of ribosomal RNA? rRNA. A. It carries genetic information. B. It catalyzes peptide bond formation. C. It binds amino acids. Or D. It unwinds DNA. Yes, that's the correct answer. It catalyzes peptide bond formation. rRNA is a major component of ribosomes and plays a catalytic role in protein synthesis, forming peptide bonds between the amino acids. It does not carry genetic information that's carried by mRNA. It catalyzes peptide bond formation. It does not bind to amino acids. The tRNA does that, and unwinding of DNA is done by helicase, not rRNA. So that's pretty much it. The questions from the questions to help you go through the assignment of fifth week. Now, uh, the fourth week answers have been released, so we'll just discuss. You know, if you have any problem, just ask me. But these answers have already been released. I'm just going through it so that if anyone has any problem, they can ask me. Which of the following types of compounds is proof of the following statement? A compound can be achiral yet still have a chiral carbon. Not a chiral carbon, still have chiral carbons. The correct answer is mesocompounds, because it has a plane of symmetry, and it has to have at least two chiral carbons, so that there can be a plane of symmetry passing through it. So, you know, that's how it becomes mesocompound. The second question was, which of the following term is used to describe the concentration of a drug required to produce measurable effect in 50% of animals tested? Yes, sir, sure. If you're done and you know this, you should you can leave. Thank you. Um, the next question is, the chemical structure of penicillin. How many chiral carbon does penicillin have? So, in order for a carbon to be chiral, it should have like four different substituents. So, let's start from uh, right to left. The carbon here has two similar substituent here as well as here. So, this is not a chiral carbon. Let's talk about this carbon here. It has one H and one carboxylic acid. It has nitrogen here and this group here. So it obviously has four different substituents. So this can be one of the uh, chiral center. Here the carbon again has like one H and this group as well as this group here. Now all the other, because this is not a symmetric structure, all the substituents are different. So again, this can be a chiral center, and the next carbon next to it, it has this group, again this group and this group along with an H. All four substituents are different, so this can be the third chiral carbon, and this is a planar structure. We don't talk about um, chirality in normal sense here. We talk about phases when we talk about uh, planar structure, E phase and C phase. So this cannot be the chiral center and not this one. So the correct answer is 3. The next question was, a certain compound X occupied a site of an enzyme exactly opposite to that of active site. This immediately resulted in change of shape of the active site. X is called a. So in case of competitive inhibitor, the inhibitor goes and binds to the active site. So it competes for the same site along with the substrate. So that's called competitive inhibitor. In case of non-competitive inhibitor, the inhibitor goes and binds to allosteric site, that is a site or other than active site. So it does not compete with the substrate But once the non-competitive inhibitor binds to the allosteric site, it changes the shape of the active site. So once the shape of the active site changes, the substrate no longer recognizes the active site and it is unable to bind there. And because it is unable to bind there, it cannot, work or catalyze the reaction. Hence, the reaction is inhibited. So the correct answer is non-competitive inhibitor. The next question was to assign correct chirality. Let's do this. So, in this, like, basically hydrogen and oxygen is like protoning molecules. So if we like, let's start from the bottom. Let's take the blue. So here, this one should be the first priority, and here's C, OH, and again then C. So 2 and 3. So you can, oh, wait a minute, let me, this one would be more clearer. So it's supposed to be R because it's leaning there, but we see hydrogen is coming out of the plane, so R would change into S. So, and what about this one? Let's use different color. The first priority will be here. The second priority will be C double bond O, 2, and this one will be 3. So it will be S, but because H is coming out of the plane, S will change into R. So it will be 2R and 3S. The next question was which of the following description best fits an agonist? A compound that has same effect of on a receptor as an endogenous chemical messenger, or B a compound that binds to a receptor and activates it but to a lesser extent than the endogenous chemical messenger, or C a compound that binds to a receptor, fails to activate it, and prevents endogenous chemical messenger from binding, or D a compound that binds to a receptor, fails to activate it, and leads to drop in inherent biological activity. So when we talk about agonist, we talk about um, uh, you know the kind of receptor, that the messenger that has same effect as the natural ligand. So option A is correct. The second one is talking about a partial agonist. The third one is again poison, and it fails to activate it. Yeah. So antagonist, and the last one is poison. Identify an amino acid in the active site whose residue could bind to a drug principally by hydrogen bonding. So you have to remember the structure of amino acids for this one. Serine, there is OH in serine. Aspartic acid has acidic uh, to it, and glycine, it's like a chiral amino acid, and valine, it's small. And it has like CH3 as its like um R. So the correct reaction, obviously hydrogen bonding is the one that will go with OH. So serine, ionic bonding, aspartic acid because it has COH, van der Waals interaction, obviously 3, and fourth glycine because it has just H. Which of the following terms best defines the intrinsic ability of a drug to bind to a receptor? An year 18-year-old girl suffering from seasonal I always struggle from saying this word. Rhini- rhinitis, rhinitis, rhinitis started a therapy with loratadine, a drug that binds to H1 histamine receptors. So when we talk about in a drug to, you know, how well it binds with the receptor, and we generally talk about affinity. That's like the basic determining definition of affinity. So yeah, the correct answer is affinity. Which of the following compounds is achiral? So, all the among all the options, just try to find a plane of symmetry passing through it. So in the first one, you see that despite having this, like, they don't have a plane of symmetry despite having like same kind of arrangement. OH is in the opposite end, and H is at the opposite end, so there's no plane of symmetry there, neither in the third one or in the fourth one. See, odd, even when you, it's a simple trick when you talk about alleles. You say odd, even, and if it was odd and then it was CH3, then it would have plane of symmetry, because then you could pass through it. But that's not the case here. It's just odd, even, like, so there is no plane of symmetry here. You can see plane of symmetry only in the two, so that's the correct answer. The next question was, partial agonist is used to describe a drug that binds to a receptor, fails to activate it, and leads to a drop in inherent biological activity. True or false? That's false, because partial agonist actually binds to it and activates it, but not to the extent that a natural ligand would. So this statement stands false. The next question was, a drug that blocks the binding site of an enzyme from a substrate are called, obviously, inhibitors. Receptors are different targets, and messengers are like CMP that actually, uh, that in the case of receptors, they actually travel with the message, and poisons are the one that destroys it. So obviously the correct answer is inhibitors. The last question was, an agonist contains an alcohol, amine, and aromatic ring, all of which act as binding groups. Which of the following modification is likely to result in an antagonist? So, converting the alcohol to methyl ether. B. Adding extra aromatic ring to the structure. Or C. Synthesizing analog which lacks aromatic ring. Or D. Converting amine to an amide. So they are talking about antagonist. So what, in all the other option apart from B, adding an extra ring to, uh, you know, uh, adding an extra aromatic ring to the structure, all the other is like playing with the binding groups of the alcohol. So even if one of them is changed, the other will still have interaction. So they can be partial agonist, if not like agonist. So it's talking about antagonist. So for that, the second option is correct. If you have any question, please feel free to ask it. If not, we can conclude this session. I'll share the slides soon and the recording of the session. So, okay. Thank you. Good night.